Good morning, everyone. Today I'm going to talk on phase 2 or the conjugation reaction. You all know phase 2 or the conjugation reaction are second type of xenobiotic metabolic reaction. In the previous lecture, we discussed in detail about the phase 1 metabolism, the enzyme involved in the phase 1 metabolism reactions that are Cytochrome P450 enzyme system, the whole family, their nomenclature, everything we discussed on the last time. We had a special focus on oxidation, reduction, and hydrolysis reaction of phase 1 metabolic reactions. So today I'm going to discuss on phase 2 metabolic reactions or the conjugation reaction. So let us have look on to the slides regarding the phase 2 or conjugation reactions. Now, phase 2 or the conjugation reaction, what are these? Conjugation is a process by which the foreign molecule and their metabolites are coupled with the conjugating agents and are converted to soluble non-toxic derivative which are easily excreted in urine. Is it clear to everyone? See, for phase 2 metabolism to happen or the conjugation reaction has to happen, there is a requirement of any entity, at least it should have one functionality or one functional group should be there. Suppose we want to conjugate the benzene, it is not possible because benzene doesn't have any functionality, no hooks are present by which the conjugating agent is going to conjugate with it. But if we are inserting the 1-hydroxy group via the phase 1 biotransformation reaction, then it becomes, benzene becomes phenol. Benzene kya ho ja raha hai? It converts to phenol. And this phenol now capable of conjugating with conjugating agents via the phase 2 biotransformation. And now you can see it can, now it has become more or easily excretable molecule. So always what are the benefits of phase 2 type of conjugation reaction? It always increases the hydrophilicity of the molecule. So conjugation reactions can occur independently or can follow the phase 1 reaction. Conjugation takes place primarily in the liver, but can occur in kidneys also because few enzymes concerned with this are present in kidneys as well. After conjugation, the products are generally rendered non-toxic, but in certain conditions, they are left unchanged or become toxic. I already told you the fate of xenobiotic metabolism, whether it is via phase 1 or phase 2 or via both. It may convert the molecule from less active to more active, from less toxic to more toxic, or from more toxic to less toxic. Okay, so metabolically inactive to active product, or sometime active to sometime inactive to active. So any kind of fate can happen of the xenobiotics once they undergo xenobiotic biotransformation. After the conjugation, the products are generally rendered non-toxic, but in certain conditions, they are left unchanged or become more toxic. Are you agreed with this term? So all the statements are clear to everyone. Should we move ahead? Now, types of phase 2 reactions. The one and more abundant kind of reaction, phase 2 reaction is glucuronidation, then sulfation reaction, then acetylation, then methylation, then conjugation with amino acid, then conjugation with glutathione. So these are the major conjugating agents present abundantly in our body, and they are all set or all time ready to conjugate with some substances which need to be excreted out of the body. Now let us first talk on to the glucuronidation reaction. Glucuronidation is the most frequent type of conjugation reaction. UDP, glucuronic acid, is the glucuronyl donor, which is formed in the uronic acid pathway of the glucose metabolism. Have you studied the glucose metabolism? It is done. If it is not done, you will see in the subsequent lectures that there is a uronic acid pathway. So uronic acid pathway is the major path for generation of glucuronides. Glucuronyl transferase is the major enzyme. Transferases are the glucuronyl transferases present in endoplasmic reticulum and cytosol, and they act as a catalyst. Means they are catalyzing all the glucuronidation reactions. The glucuronide may be attached to oxygen, sulfur, nitrogen groups of the substrates. You can see the example. Glucuronidation because bilirubin, though it is generated endogenously, I already told you in the slides, the maximum xenobiotics are exogenous, but there are few who have the same fate as those of exogenic xenobiotics. They are also included in the category of xenobiotics. Say, for example, your bilirubin. Then you have a steroids. Steroids hormones, they can also undergo glucuronidation reaction most frequently. So have a look on UDP glucose, and this is the UDP glucose, and it is converting. I'm using this laser pointer. So this UDP glucose, you can see the UDP glucose dehydrogenase, or is the enzyme. It is converting UDP glucose into UDP glucuronic acid. This is one of the important reaction of uronic acid pathway. And in this course of conversion, there is a requirement of 2-NAD, and 2-NAD get reduced to 2-NADH. So glucose is converted to glucuronic acid as an adduct of UDP. In the next step, you can see the UDP glucuronic acid. Now it is all set to conjugate with bilirubin, and bilirubin has also got one hydroxy group. So through hydroxy group, there will be a conjugation reaction, and the enzyme will be UDP glucuronyl transferase, and this way it is going to be converted to bilirubin monoglucuronide, and UDP is now released, and it can be recycled and reconverted to UDP glucose, and it can facilitate the reaction again and again. Next, that was the then bilirubin monoglucuronide with another molecule of UDP glucuronic acid, it can convert into the bilirubin diglucuronide, and which is highly water soluble, and that can undergo excretion via feces or via urinal, because it has become now more hydrophilic. Now it is in the excretable form, and UDP is again released. So most of the bilirubin excreted in the bile of mammal is in the form of bilirubin diglucuronide. Bilirubin UGT activity can be induced by a number of clinically useful drugs, including the phenobarbital. Phenobarbital is one such drug which induces the enzymes concerned with the formation of bilirubin diglucuronide. So like glucuronyl transferases activity is induced by frequent use of phenobarbital. It is a CNS active drug which has induction capability. It can induce the activity of this enzyme. Now, second important reaction is sulfation. What is a sulfation? Sulfation is the addition of sulfate molecule. You all know sulfur contains 4 oxygen, SO4, and these are highly electronegative. So to whatever entity they are going to attach or they are conjugating, they will convert the molecule into more hydrophilic entity. So have a look on this. Sulfate donor is adenosine 3-phosphate 5 phosphosulfate. In brief, we call this PAPS. PAPS or PAPS is always the donor of sulfur group. In all, not only in this reaction, in most of the sulfation reactions, the PAPS is always the donor of sulfate group. And this compound is called the active sulfate. In this form, it can donate the sulfate to any of the molecule in course of various biochemical reactions. So the enzyme is sulfotransferase, similar as in a similar manner as in the previous reaction you have seen, the enzyme was glucuronil transferase. Here the enzyme is sulfotransferase. The compound which are conjugated with sulfate are as follows. Phenols can undergo sulfation, cresol, indol, and steroids, and then estrogen, androgens. All these molecules can nicely undergo sulfation reaction, and these entities, after being sulfated, becomes more hydrophilic and less active, and they can easily be excreted out of the body. So you can see all this group of molecules or these chemicals, all of them contain one functionality, especially in the form of hydroxy group. Because conjugation ke liye you need some hooks to attach with. So always the one functionality, maximally in the form of hydroxy group, it can be in the form of amino group, it can be in the form of other thiol groups also. Tyrosine to form tyrosine O sulfate, which is required for formation of fibrinogen. Another reaction is glycosaminoglycans and glycolipids and glycoproteins are also synthesized via O-sulfation reactions. You can see the example of, this is the phenol. This is which molecule? This is a simple phenol. You would have seen in your previous, you are very well versed with this phenol molecule. So active sulfur, what is active sulfur here? PAPS. PAPS is a donor of sulfate group. So PAPS is here and it is converting this molecule. The hydroxy group is now OSO3H, means sulfated now. It is phenyl sulfuric acid. So sulfotransferases are localized in the cytosol and transfer the sulfate moiety mainly to the hydroxy group. The donor of sulfate is PAPS, and which is synthesized from 2 molecules of ATP and 1 mole of 2 moles of ATP and 1 mole of sulfate. Is it clear to everyone? Next kind of reaction is acetylation. Acetylation is represented by, suppose X is a, what is X? X is a xenobiotic, and the donor of acetyl group is acetyl coenzyme A. This is the acetyl coenzyme A, very versed molecule. It is pivotal to the various energy releasing reactions you would have seen previously in the Krebs cycle, or pyruvate converts to acetyl coenzyme A. Acetyl coenzyme A is also the starting material for fatty acid synthesis, for cholesterol, for ketone bodies, and acetyl coenzyme A also is utilized for various metabolic reactions. So you can see here it is acting as a donor of acetyl group. So this is a xenobiotic. So in course of conversion, it becomes now acetyl X, or xenobiotics now conjugated with acetyl group, and coenzyme A is released. So where X represents a xenobiotics. Acetyl coenzyme A is an acetyl donor. These reactions are catalyzed by acetyl transferases. Okay, so there is a, no, you put the name of the conjugating agent and plus transferases. Sulfotransferases, then glucuronil transferases, and here it is acetyl transferase. So present in the cytosol of the various tissues, particularly in the liver, polymorphic types of acetyl transferases exist, resulting in individuals who are classified as slow or fast. So various polymorphic forms are generated, few are fast acting, few are slow acting. Then slow acetylators are more subject to certain toxic effects of drugs because the drug persists longer in these individuals. Now, you can see the example of this acetylation. The compounds conjugated by acetylation are sulfonilamide, PABA. PABA is a para-amino benzoic acid, then isoniazid. It is an antitubercular drug. So here it is the PABA, para-amino benzoic acid. So acetyl coenzyme A is condensing here and is conjugating with NH. Amino group is now converted to N-acetanilide. So this aceto group is now protecting the amino group. It has become NHCOCH3. So it is acetylated para-amino benzoic acid. The enzyme is, which enzyme is working here? Name the enzyme, please. Very good. Acetyl transferase is the enzyme. The next type of reactions are methylation. Methylation is happens in a very limited manner in the body, and SAM or S-adenosyl methionine active, it is known as active methionine, act as a donor of methyl group. In brief, it is written as SAM. You can note it down. It is SAM. It is SAM. S-adenosyl methionine. The reactions are called transmethylation reaction, and enzymes catalyzing the reactions are methyl transferases. You can see the example of methylation reaction. Compounds conjugated by methylation are nicotinamide, and nicotinamide is converted into N-methyl nicotinamide. So here the donor will be SAM or S-adenosyl methionine. Methionine, what is methionine? Methionine is an essential amino acid. Essential or non-essential amino acid? Sure? Nicotinamide, now it is converted to N-methyl nicotinamide. So then para-methyl aminoazobenzene can also be converted into para-dimethyl aminoazobenzene. It acts as an hepatic carcinogen. So then O-methylation of estrogen, norepinephrine, endogenous reactions are, these are the endogenous reaction happening in vivo. Estrogen to methylestrogen, then norepinephrine, epinephrine and their metabolites, they require O-methylation reactions. The next is conjugation with amino acid. So the main conjugating agent here, it is in glycine. Glycine is one amino acid which is achiral and which frequently undergoes the conjugation reaction. We can see the example of benzoic acid. It is converted into the hippuric acid with the glycine, and the reaction here, what will be the enzyme? Enzyme will be glycinyl transferase. Okay, the enzyme will be glycine transferase. And this, the first reaction is very, very frequently happens among the horses. And this reaction, like in their urine, horse urine contains abundant quantities of hippuric acid. And whatever like eatables he eats, it is rich in benzoic acid, and benzoic acid is excreted out of the body via glycine, via glycination, conjugation reaction, and it turns to hippuric acid, which is highly excretable, and it can easily be removed out of the body through urine of horse. Then nicotinamide, it can also conjugates with glycine, leading to the nicotinuric acid. Then cholic and deoxycholic acid. What are the cholic and deoxycholic acid? Anyone can tell? Have you heard these names? I taught you when I was discussing on cholesterol and its uses, its derivatives. The products derived from cholesterol. So cholic acid and deoxycholic acid. What are these? These are bile salts. These are bile salts. Keep remember, you're not going to forget the things which are being taught previously. have vigorous revision. So the cholic and deoxycholic acids are conjugated to form glycocholic acid and glycodeoxycholic acid. The next is conjugation with glutathione. Glutathione is a small peptide which does consist of, it is a gamma glutamyl cystenyl glycine. 3 amino acids are involved in the synthesis of glutathione. It's another name for glutathione is gamma glutamyl cystenyl glycine. You can note it down. The enzyme catalyzing these reactions are called glutathione S transferase. A variety of glutathione S transferases are present in human tissues. They exhibit different substrate specificities and can be separated by electrophoretic and other techniques. If a potentially toxic xenobiotics were not conjugated to glutathione, they would be free to combine covalently with DNA, RNA, or cell proteins and could lead to disastrous impact onto the cell, and they will finally destroy the cell. The GSH is therefore an important defense mechanism against certain toxic compounds such as drugs and certain carcinogens. This is very, very important reaction for detoxification of certain substances which necessarily undergo glycination reaction. As in the form of glycine conjugates, they can easily be excreted out of the body. Now, overall, in nutshell, this is what can be the effect of xenobiotics. So this is a xenobiotic. What can be the different fate of xenobiotics? Have a look onto the slides and try to understand. This is a xenobiotic via cytochrome P450 system, enzyme system, which are the integral component of phase 1. These are the phase 1 enzyme. And they convert into some reactive metabolite. Understood? Reactive metabolite means the metabolite which is having some functionality, and this functionality would have come from phase 1 reaction via oxidation or via hydroxylation or via reduction. These are the major reactions which lead to addition of some functionality onto the xenobiotics in the presence of enzyme cytochrome P450. And next, what can happen? There is a covalent binding to macromolecule. So this reactive metabolite is very, very reactive, and it can covalently bind to the various macromolecules present in the body. In the cell, they can be DNAs or RNAs, they can be some proteins or enzymes or the hormones or some glycoproteins. So they can covalently bind. They have the capability because इनके ऊपर एक ऐसा functional group है, and because of that functionality present, they are highly reactive. They may cause cell injury. They may form haptens. They may cause mutation if they are going to wind up with the DNA. And then haptens, haptens once they are formed, they are behaving like antigens. So there is a production of number of antibodies against them. And then there is a sort of defense mechanism gets started, it gets stimulated, and that leads to cell injury. So that these can be the following fates of phase 1 metabolism if the reactive metabolite is not being controlled. So it will have positive or negative, both kind of impacts. If it is intentionally being prepared against some type of cell which are to be destroyed. So these are the mechanism through which it can destroy those cells. Suppose as some cancerous cells are to be destroyed, so a special metabolite is to be generated via phase 1 metabolism. Some drug is being designed in a way, and now that is going to specifically kill the cancerous cell. Okay. Then reactive metabolism, what can be the other fate? Other fate can be like there can be conjugation reaction, and conjugation reactions via glutathione transferase or via a number of conjugating enzymes and reactions. It can convert it into the non-toxic metabolite in most of the cases. But the thing, what is the quality of that adduct, the conjugate which is being produced is highly excretable, because ये जितने भी आपके conjugating agents हैं, maximally they are producing, they are inculcating some hydrophilicity, additional hydrophilicity in the phase 1 metabolite or in the xenobiotics, which may skip the phase 1 metabolism. Understood? So it may convert into the non-toxic metabolite, which is easily excretable. So that's all about the phase 2 metabolism. Understood? Any of the points you are not able to understand, you can ask me. One thing, let me clear you, any xenobiotic which enters your body, if already it has some functionality, it may skip phase 1 metabolism. Not necessarily it has to necessarily undergo phase 1 metabolism. It is already hydrophilic. It has some some electronegative functionality which are highly hydrophilic. Then it doesn't require any phase 1 metabolization. It may directly undergo phase 2, or it can skip phase 2 as well. It depends upon the circumstances, the requirement of the body. If it is not stagnant, easily excretable, so it will, after performing its function, it will go of its own accord. That time there is no interference. There may not be any interference of phase 1 or phase 2. Understood? Clear to everyone? So with this, I am ending up this lecture. If you have any other questions, you can ask us.